Welcome to the PubMed Daily Review for April 15th, 2026. Today we scanned 7,210 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Therapy-induced cholesterol biosynthesis drives lung cancer dormancy and drug resistance by Yikai Zhao and Hongbin Ji from Key Laboratory of Multi-Cell Systems, Shanghai Institute of Biochemistry and Cell Biology, Center for Excellence in Molecular Cell Science, Chinese Academy of Sciences, Shanghai, China. Let's break down its key contributions. Lung cancer-targeted therapies have revolutionized treatment, yet a persistent challenge remains: incomplete responses and eventual relapse. This often stems from cancer cells entering a dormant state, where they survive therapy but don't proliferate, making them incredibly difficult to eradicate. Understanding how this dormancy is triggered and maintained is crucial for developing more effective, lasting treatments. To unravel this mystery, researchers employed a multifaceted approach. They utilized various lung cancer cell lines, including those with common EGFR, KRAS, and ALK mutations, alongside xenograft mouse models. Crucially, they also analyzed human lung cancer specimens before and after targeted therapy. A wide array of techniques, from advanced sequencing to genetic manipulation, allowed them to meticulously track cellular changes and signaling pathways under drug pressure. The findings were striking. They discovered that molecular-targeted therapy, surprisingly, induced cholesterol biosynthesis in cancer cells. This increase in cholesterol was directly linked to cells entering dormancy and developing drug resistance. However, when statins were combined with targeted therapy, they effectively blocked this cholesterol synthesis, preventing dormancy and leading to dramatic tumor regression. Further investigation revealed that the PERK, EIF2 alpha axis of the unfolded protein response was a key driver of this therapy-induced cholesterol production, which in turn reactivated AKT signaling to promote cell survival. Ultimately, a triple targeting strategy, combining inhibition of the oncogenic driver, cholesterol biosynthesis, and aurora kinase, almost completely eradicated the cancer cells. This study uncovers a critical, previously unrecognized mechanism. Targeted therapy inadvertently triggers cholesterol biosynthesis, which then acts as a survival program, pushing cancer cells into dormancy and fostering drug resistance. The good news is that combining existing targeted therapies with statins, common cholesterol-lowering drugs, shows immense promise in preventing this dormancy and improving treatment efficacy. Looking ahead, the innovative triple targeting strategy, hitting the oncogenic driver, cholesterol synthesis, and aurora kinase, presents an exciting new avenue for achieving more complete and durable responses in lung cancer patients. Up next, our second paper out of 5. Human stem cell-derived neurons establish functional inhibitory-excitatory cortical circuits in a chimeric transplantation model by Cameron P., J. Hunt, and Carlos W. Gantner from Florey Institute of Neuroscience and Mental Health, Melbourne, VC, Australia. Let's break down its key contributions. The brain's intricate balance of excitation and inhibition is fundamental for healthy function. When this balance is disrupted, it can lead to various neurological conditions. While we've made strides in replacing lost neurons, most approaches focus on excitatory cells. This study asks a crucial question: can we create functional circuits by combining both inhibitory and excitatory human stem cell-derived neurons? To tackle this, researchers developed a sophisticated model. They generated human stem cell-derived inhibitory neurons from the medial ganglionic eminence and excitatory neurons from the cortex. These were then co-grafted into mice. Using optogenetics, they could precisely stimulate the inhibitory neurons and then assess their impact on the excitatory neurons, looking for functional synaptic connections. The results were compelling. They found that the grafted inhibitory neurons were indeed functionally integrated. When stimulated optogenetically, these inhibitory neurons successfully induced inhibitory postsynaptic potentials in the excitatory neurons. This effect was confirmed when it was blocked by gabazine, a specific inhibitor. This demonstrates that these stem cell-derived neurons can form functional circuits. So, what does this mean for the future? This study provides crucial proof of concept that we can engineer functional neural circuits using stem cell-derived neurons. This is a significant step towards developing more effective cell replacement therapies, particularly for conditions like stroke where restoring the excitation-inhibition balance is vital. Furthermore, this model offers a powerful new platform for studying complex neurological disorders in vivo. Up next, our third paper out of 5. Cell clustering promotes a metabolic switch that supports metastatic colonization by Christian F. Labuschagin and Karen H. Vuzden. Let's break down its key contributions. Today, we're diving into a fascinating study that explores a critical aspect of cancer progression: metastasis. The core question this research tackles is how cancer cells adapt their metabolism, especially when they cluster together, to successfully spread and form new tumors. This is incredibly important because metastasis is responsible for the vast majority of cancer deaths, making any insight into its mechanisms a potential game-changer for patient treatment. This basic science study likely employed a range of sophisticated techniques to unravel these complex cellular processes. Researchers probably utilized various cancer cell lines, potentially in advanced 3D culture systems, to mimic the in vivo environment more closely. They would have performed detailed metabolic profiling to identify changes in energy pathways and conducted functional assays to assess metastatic potential, all aimed at understanding how cell clustering drives these critical metabolic shifts. The key findings from this research are quite compelling. The study demonstrated that cell clustering isn't just a physical phenomenon. It actively promotes a significant metabolic switch within cancer cells. This metabolic reprogramming, in turn, was shown to be critical in supporting the cell's ability to colonize new sites and form metastases. Interestingly, the research also highlighted that detached cells, those on their journey to new locations, become particularly dependent on glycolysis for their survival, a crucial insight into their resilience. So what's the big picture here? This study provides compelling evidence that cell clustering isn't just a passive event. It actively drives metabolic changes that are essential for cancer cells to metastasize. This opens up exciting new avenues for therapeutic development, suggesting that by targeting these specific metabolic switches, especially glycolysis in detached cells, we might be able to disrupt the metastatic process. Of course, the next crucial step will be to translate these fundamental insights into effective clinical strategies and validate them in human studies. Up next, our fourth paper out of 5, Human-Centric Modeling in Metastatic Breast Cancer, Organoids, Organ-on-Chip Systems, and New Approach Methodologies in the Post-FDA Modernization Act 2.0 Era by Hissa Alatawi and Satya Narayan from Department of Physiology and Aging, College of Medicine, University of Florida, Gainesville, Florida, 32610, USA. HHN, EV. Let's break down its key contributions. Metastatic breast cancer remains a devastating disease, largely because it's so complex and often resistant to treatment. For decades, we've relied on models that just don't fully capture the nuances of human tumors, leading to many drugs failing in clinical trials. But now, there's a significant shift happening, with new regulations supporting more human-centric approaches. This paper explores how advanced models like organoids and organ-on-a-chip systems, combined with AI, could revolutionize how we test drugs and predict patient responses. This paper is a review examining the latest advancements in human-centric models for metastatic breast cancer. It focuses on patient-derived organoids and organ-on-a-chip systems, highlighting how integrating these with artificial intelligence is key. The core idea is that these advanced platforms can more accurately mimic the complexity of human tumors and their microenvironments, offering a better way to test drug effectiveness compared to older methods. The results are quite striking when you look at the limitations of current methods. Traditional animal models, for instance, have shown only about a 9% concordance rate with actual clinical trial success. This highlights a massive translational gap. On the other hand, AI integration in these new models is achieving high technical metrics, like segmentation accuracies over 90%. Crucially, regulatory bodies like the FDA are now actively supporting these new approach methodologies, paving the way for their use in drug development. So what does this all mean for patients? These human-centric models represent a significant leap forward, offering a more accurate way to understand and treat metastatic breast cancer. The integration of AI is critical for making these complex systems reliable and reproducible. With regulatory bodies now on board, we're moving closer to a future where these advanced platforms can help predict treatment responses more effectively, ultimately improving outcomes for patients. Up next, our fifth paper out of 5, long non-coding RNA-derived peptides as a novel source of tumor neoantigens, expanding the Immunopeptidome beyond canonical coding regions by Ismael Lopez Calvo and Ángel Vizoso Vázquez from Centro Interdisciplinar de Química y Biología, SICA, Universidad de Coruña, Campus del Vina, as Carboliras, S/N, 15071A Coruña, Spain, ILC, IBC, SMR, CRS, AFG, JMPM, ISS, AM, MLM, MEC. Let's break down its key contributions. Cancer immunotherapy has revolutionized treatment, but its effectiveness is often limited in tumors with low mutational burden. This is because these tumors have fewer of the classical mutation-derived neoantigens that T cells can recognize. Recent research suggests that the pool of tumor antigens is much larger than previously thought, extending beyond protein-coding genes. Specifically, peptides derived from long non-coding RNAs, or LNCPPs, are emerging as a promising new frontier. This paper is a comprehensive review that synthesizes the latest research on long non-coding RNA-derived peptides, or LNCPPs. The authors explore the molecular mechanisms behind how these peptides are generated, including translation from small open reading frames within LNC-RNAs. They also discuss the experimental and computational techniques used to identify these peptides, such as translatomics and immunopeptidomics. The review focuses on how these LNCPPs can be processed and presented by the immune system, potentially serving as novel tumor neoantigens. The review highlights several key findings. Firstly, LNCPPs have demonstrated the capacity to elicit CD8 plus T cell responses, meaning they can be recognized by the immune system as foreign. Importantly, these peptides often exhibit high tissue and context specificity, making them potentially more selective targets. This specificity is particularly valuable for expanding the range of actionable antigens in tumors that typically have a low mutational burden. Overall, the evidence strongly suggests that LNCPPs are a significant, yet largely untapped, source of tumor neoantigens. The clinical significance of this research is substantial. By identifying peptides derived from LNCPPs, we gain a new strategy to potentially enhance cancer immunotherapy, particularly in tumors that haven't responded well to current treatments. However, there are challenges ahead, including the need for more robust methods to identify and validate these peptides' immunogenicity. Future work will focus on overcoming these hurdles to translate this knowledge into effective personalized cancer vaccines and T-cell therapies. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.